Assalamu alaikum and hello everyone. Hopefully you all are doing well. Today we will discuss about the sorry relative importance of bonding interactions. Previously we were discussed about the structure of proteins. The most significant kind of interaction those are present in tertiary structure of protein. Right? In tertiary structure of protein, we have different kind of a bondings. It could be covalent bonding, ionic bonding, hydrogen bonding, or van der Waals forces. Right, I just wrote it according to their strength. Covalent is the most stronger one, ionic is less than covalent, then hydrogen bonding, and then van der Waals forces. But their importance is quite opposite. Van der Waals forces are the most significant one in terms of tertiary structure, and then the hydrogen bonding, and then ionic and the covalent bonding is least important. There are two main reasons because there are more opportunities for van der Waals forces and the hydrogen bonding than the covalent ionic bonding. Only few amino acids if you just consider the covalent bonding, very few amino acids go for the disulfide linkage or disulfide bridges. Right, but there are certain proteins those are rich in disulfide bridges, right? In that particular case, the covalent linkages are more significant in terms of their structure, right? But generally, disulfide linkages are more significant in small peptides. And the small peptides, best example is hormones, right? We can take the example of two hormones, oxytocin and vasopressin, right? If you just write their sequence, right, one of the easiest ways to recall this like CTP GACP, right? This carbon will develop this disulfide linkage. This is the vasopressin. The second one is oxytocin. In terms of oxytocin, it's CTIGAC, right? Then this carbon will develop a bond with these two sulfurs and disulfide linkage will develop, right? These are the examples of two hormones those are having disulfide linkages, right? And the next point is the formation of disulfide linkage, right? Usually it is formed in endoplasmic reticulum, right? Their mode of formation is through oxidation, right? And how the oxidation occur, we need oxidants. Two most common kind of oxidant is one is the oxygen and second one is hydrogen peroxide, right? If you consider the reaction, right, if when two thiols react together, we end up with disulfide linkages with the release of two protons with two electrons, right? But when sometimes it happens that this disulfide bridge will react with one another and just mark it as a prime, R prime, right? Thiol, then resultantly it can replace the previous R group, alkyl group, and replace it, right? This could be the other possibility to have a disulfide linkages, right? The next point is about the right. If you can consider the structure, then disulfide linkages are least important or they play minor role in controlling the tertiary structure of any protein, right? If you consider the ionic bonding, We have discussed about the covalent. In terms of ionic bonding, they have very limited amino acids, they go for the ionic bonding, right? Usually amino acids are having their residues attached with them, they go for the ionic bonding, right? And most of the time they also preferably have a multiple number of hydrogen bonding in the van der Waals forces, right? The second main point is why the van der Waals forces and the hydrogen bonding is more significant in terms of tertiary structure is proteins are usually surrounded by water, right? And water is highly polar, right? So it can readily interact with polar substances or hydrophilic substances, and the residues, those are able to have a hydrogen bonding. These are the three main possibilities that the protein can interact with the hydrophilic environment, right? While the non-polar, hydrophobic amino acids and its residues, they cannot interact with water, right? Okay, the next point is stability of structure. Okay, just to attain the stability of structure in terms of protein, what happened that most hydrophilic groups are present on the surface of a protein, right? Because it is surrounded by water, so they can interact with the water, right? And the most hydrophobic, these are the hydrophilic, hydrophilic on surface, right? Hydrophobic are present in the center of proteins, right? Just to avoid the water, and but they can interact with themselves, with each other, the hydrophobic components, right? Hydrophilic amino acids, they form hydrogen bonds, hydrogen bonding with water, and they contribute mainly towards the tertiary structure of protein. So hydrophobic and the van der Waals interactions mainly determine the three-dimensional shape of proteins. All the hydrophilic amino acids, they are busy to have interactions with the water which is surrounded by it, right? So they are actually not contributing towards structure as such, right? The most significant one is towards the structure is the hydrophobic interactions, which is present inside in the center of the protein, right, to determine the three-dimensional structure of protein. If you consider the center of protein, that central protein is non-polar, if any globular protein, or it is hydrophobic, which results to have multiple applications of it. Number one, it helps like enzymes to play their role in non-aqueous medium, like there's particular reaction those require non-aqueous medium, so that's why the core of the enzyme of any any protein can act in non-aqueous medium because of its hydrophobic nature and the non-polar nature, right? That enable the enzymes that they can catalyze the reactions. Those are impossible in aqueous medium in our body, right? And because enzymes contain a specific hollow cavity, right? That particular region where the enzyme can interact with the protein is, or any drug or anything like, so they are containing that particular site which is interacts is called active site, right? And it's basically in the center and it is hydrophobic. It provides a non-aqueous medium for the reaction, right? While the protein similarly contain a site which is called binding site, right? And that's also like a cavity, right? And it's like a hollow, right? And it's normally available for the natural ligands which is present in our body, right? So it enables the natural ligands to interact, right? And so the van der Waals forces and the hydrophobic interactions play important role in binding of the ligands, right? And which is important to design any drug to target the binding site, right? So whenever we're designing a drug, we need to consider such kind of a properties and the features of proteins, right? Then if you consider the conformation of peptide bond, right? Then peptide bond could be cis or trans, that's obvious, right? But the most common one is a trans one, right? If I will give you the example, like this is the alpha carbon, this is also the alpha carbon, right? They both are opposite, so it's a trans structure, right? In terms of cis, right, it's alpha carbon, right? When both is alpha carbons on the same side, then it's a cis structure, right? But generally we have a trans structure, but mostly we have a trans structure, right? With the the restricted bond orientation, right? So the number of possible conformation is restricted in terms of its structure, right? Okay, so then the most important factor is to consider the entropy. What is entropy? Entropy is the disorderness, right? So what we want, like any protein without polypeptide bonds cannot fold, right? When the folding occur, then entropy enhance means disorderness enhance, right? Otherwise there is a particular sequence and pattern and they align themselves, right? So we want to change the entropy, which is very much important to require to form a ordered structure, is extremely unfavorable, right? So we want to have a folding, right? So peptide bonds also form hydrogen bond with amino acid chains and play important role in structure determination, right? So it's very much important to have a folding and to change in entropy. Okay, any other point regarding this? I guess I've done with all the points. And if you have any confusion, you can ask. Thank you.